We are done with the first two parts of the central dogma, which were DNA replication and transcription. So now we're down to the last part, which is the last step in the creation of the gene product. So we learned about DNA replication, which takes place during the cell cycle or when a cell is trying to produce an offspring. So it needs to produce an exact copy of its gene. So that is the purpose of DNA replication. And then we also tackled the process of transcription, which is the creation of an mRNA transcript from a gene. So remember that not all of the DNA sequence in an organism is considered a coding sequence. So only those with a particular gene product, which is what we call a gene, those are the ones that are transcribed. And now we will be focusing on translation, which is decoding of the message that is carried by your mRNA transcript. So the flow of information from your blueprint, which is stored in your DNA, is such wherein you have the coding strand in your DNA. So this coding strand has a complementary strand, which is what we call the template strand. And then the template strand is read by your RNA polymerase, and it synthesizes a complementary mRNA transcript. So recall that the sequence of your mRNA transcript is similar to the sequence of your coding strand. The only difference being that the T in your coding strand is replaced by a U in the mRNA transcript. Now, this message transcribed in your mRNA is finally translated to the gene product, which is a polypeptide, or essentially a protein, which is a string of amino acids. So, this process of translating the code that is stored in your DNA revolves around codons. So, the code is actually a nucleotide triplet. So if you have an mRNA transcript, you divide this mRNA transcript into triplets. Each triplet or each three consecutive nucleotides correspond to one codon. And each of this codon is translated to a specific amino acid. So that's how we create the gene product. Now let's talk a bit about protein synthesis. So protein synthesis is very important inside the cell, and this is reflected by some of the facts listed here. We know that protein synthesis takes place or is brought possible by the ribosomes, which are essentially ribonucleoproteins or complexes made up of RNA molecules and proteins. These ribosomes are found in the rough endoplasmic reticulum. So when we talk about protein synthesis, there are 70 different ribosomal proteins involved in this process, 20 or more enzymes, dozen or more auxiliary enzymes. And of course, just like what we learned in DNA replication and transcription, there are also protein factors involved in the initiation, elongation, and termination of protein synthesis. And once you have the protein product from your mRNA transcript, the story doesn't end there. So there are 100 or so enzymes that are involved in the processing of the protein that you have synthesized. And aside from this, we also have the tRNAs and the rRNAs that are involved in this process. So there are at least 40 of these. So the endoplasmic reticulum, or the rough endoplasmic reticulum to be more specific, is actually sort of connected to your nuclear membrane. So this is in the context of eukaryotes. So transcription, eukaryotic transcription takes place inside the nucleus because that's where you have your DNA. And the mRNA transcript produced during transcription should reach the ribosomes that are found in the rough endoplasmic reticulum. So these mRNA transcripts are transported from the nucleus towards your rough endoplasmic reticulum. That is why this RER is in proximity to your nucleus. Protein synthesis consumes 90% of cellular energy, and as I mentioned, proteins are the workhorses of life. They are the ones responsible for the various processes, biochemical reactions taking place inside the cell, and these reactions are important so that life is maintained. Hence, the percentage of energy required by this process. So in total, there are 15,000 ribosomes, 200,000 tRNA molecules, and including protein factors and enzymes. If we take into account all of these, that is equal to 35% of the cellular weight in bacteria. And again, what we are trying to highlight here is the magnitude or the importance of protein synthesis. Now, if we look at the rate of protein synthesis, your ribosome can synthesize a 100 amino acid protein in 5 seconds. And if we compare this with DNA replication and transcription, this is relatively slower, right? So if you recall, your DNA polymerase 3 for bacteria can, so we're talking about processivity, right? It can incorporate at least 500,000 nucleotides per second. Meanwhile, for um, RNA polymerase, it can incorporate 10,000 to 100,000 nucleotides per second. And I think we shouldn't be questioning the processivity of the enzymes and the ribosome for protein synthesis in this case, because these values are already optimized through billions of years of evolution. Okay, so we have the high processivity for DNA replication because we are trying to replicate longer sequences, right? Likewise, the processivity decreases for mRNAs because we are transcribing shorter segments. We are just transcribing genes, right, which are segments of your DNA. And downwards into the process, we have the translation or protein synthesis. So protein synthesis are also smaller in size, hence the lower processivity rate. And this process, protein synthesis, is actually balanced by protein degradation. So the only time when you need a certain protein, that's the only time that you need to transcribe the code in the gene, and then you get your protein. And once it's done with its function, the cell degrades it. Okay, so it undergoes degradation, and all of the amino acids used to make that protein can be recycled to make a different protein that is needed by the cell. So there are three important discoveries that led to the understanding of protein synthesis. So the first is the discovery where protein synthesis takes place inside the cell. So this is in the context of eukaryotes again. Paul Zamachnik used radio-labeled amino acids. So what he did was, so he has his cells and he fed these cells with radio-labeled amino acids. So these radio-labeled amino acids will eventually be used by the cell in protein synthesis. So what he did was, if he allowed um, a long incubation time, he found out that these radio-labeled amino acids are distributed all throughout the cell. So this is for longer incubation time with the radio-labeled amino acids. So time equals 0 is when he introduced the radio-labeled amino acids into his cell culture, and then he has his incubation time. So if he, let's say, observed or studied his cell culture after 4 hours, let's say 4 hours, his observation is that the radio-labeled amino acids are distributed all throughout the cell, even in its organelles. However, when he shortened his incubation time, so let's say he studied his cell culture 30 minutes after introducing the radio-labeled amino acids, what he discovered was that these radio-labeled amino acids were concentrated in a certain subcellular fraction. So he did fractional centrifugation, and he discovered that these radio-labeled amino acids were concentrated in the rough endoplasmic reticulum. And more specifically, they are concentrated on certain spots in the rough endoplasmic reticulum. And that's when they were able to establish that protein synthesis takes place in the RER, and more specifically in the ribosomes. The next discovery is, so there's a figure here that shows these fiber-like structures inside uh, a eukaryotic cell. So these fiber-like structures are the layers of your rough endoplasmic reticulum. And if you notice, there are dots lining these fiber-like structures. So those dots are the ribosomes, and they are giving off the signal because they are yeah. So this only shows the rough endoplasmic reticulum with the ribosomes attached to it. Okay. The second discovery is the association of amino acids with RNA molecules. So the same guy, Paul Zamechnik, and another scientist, Malon Hoagland, uh, discovered that amino acids are activated in the presence of ATP in the cytosol, and these amino acids get attached to tRNAs. At this point, tRNA, the tRNA molecule, was discovered by a different guy. So what they did was they had this cellular extract. So they extracted the contents of a eukaryotic cell. So they basically just have the cytosol, and they added their amino acids. I not sure, but I think these amino acids are also radio labeled, and then they were able to track these amino acids getting attached to tRNAs. And this reaction, this attachment of your amino acids to tRNAs, are not possible without ATP. So this is an ATP dependent reaction. And the last is the proposal from Francis Crick for the existence of an adaptor molecule. So according to Francis Crick, there should be an adaptor molecule that reads the nucleotide sequence of your mRNA transcript, and this same molecule also brings the corresponding amino acid. And of course, at this point, we already know that that is your tRNA or transfer RNA. So translation is not possible without the genetic code. So the genetic code um, was first thought of as a one-to-one correspondence between your nucleotides and your amino acids, but that is not possible because you only have 4 nucleotides versus 20 common amino acids. Now, if you set the code to be a duplex, so it's like you need two nucleotide coding for one amino acid. So that means there are 16 possible combinations. So that's 4 times 4, right? But this is still not enough for the 20 common amino acids. So finally, they hypothesized that the code is probably a triplet of nucleotides because that would give us 64 codes. That's 4 times 4 times 4. And that is more than the 20 common amino acids that we know. So with that, they tried to establish or find the answer to the question. Okay, so how many nucleotides are there in one code? We will later on see the experiments that were performed that established that the codon or the genetic code is actually composed of 3 nucleotides. But first, let us answer the question. So, say that we have established that the genetic code is made up of 3 nucleotides. The next question that we should ask is if the code is non overlapping or overlapping. So here we see the difference. If it's a non-overlapping code, you're basically reading 3 nucleotides at a time continuously. But if it is an overlapping code, uh, there are nucleotides that are shared by more than 2 codes. So for example, this second nucleotide U is shared by the first codon, the second codon, and the third codon. And because of that, there is limited flexibility, so to speak. Okay. Because if you have a non overlapping code, then you know that each code is unique in itself, right? So it's AUA, it's CGA, and so on. So the series of experiments that they performed to discover the genetic code also established that this code is a non overlapping one. Okay. So what they did was the nature of the experiments would be we will not be discussing the specific experiments, but we will just have a general overview. So what they did are what we call insertion deletion experiments, wherein they tried to insert one nucleotide in the mRNA transcript, or they tried to delete one nucleotide. Okay, because if it is a non-overlapping code, it's very easy to see that if you insert or delete, you should get a totally different gene product because you disturbed the subsequent codons from the position where you perform the insertion and deletion. As you can see here, if you try to insert C, then you affect the third codon and so on. And if you delete the fourth nucleotide G here, you affect the second codon and so on. So you will get a different protein product for each setup. And then if you perform insertion and deletion at the same time, like for example, you inserted A here and you deleted, it's a GCC, you deleted U. Okay, so you deleted U. The only codons affected were the second and the third codons, but the rest was fixed back into position. So the reading frame is restored here. So you will only get anomalies in the second and third codons. So you can imagine how such experiments will affect if the code is overlapping. Okay, so I mentioned reading frame. This term is very important when you're trying to study genes. So when we say reading frame, this is the way of dividing the sequence of nucleotides into a set of consecutive non-overlapping triplets. So if you have a certain mRNA transcript, there are 3 possible reading frames. So let's say we have this segment of the mRNA transcript. You can read the codons as UUC, UCG, and so on. Or the second reading frame would be you have a codon UCU, CGG, and so on. And then the third possible reading frame is CUC, GGA, and so on. So only one of these three possible reading frames for each gene is translated. The other two will not lead to a gene product. So later on, as we go into the details of translation, you will see how the reading frame is set. So the cell is not confused which reading frame it should use. Okay, so now let's go to the experiments that were performed that established the genetic code. The first experiments were performed by these two guys, and it is actually a very elegant design. So, what they did was they prepared synthetic mRNA molecules. So, the first setup was they had a poly U mRNA, and then they allowed the RNA polymerase and other important molecules and enzymes to translate this synthetic or artificial mRNA. And they analyzed the protein product, and what they obtained was a polypeptide that contains only phenylalanine. So at this point, if the code is a triplet, that means it's UUU for phenylalanine. So they also did the same for C. So they created an artificial mRNA, poly-C mRNA, got it translated, and they got a polypeptide composed of only proline. They also did this for poly A. So for poly A, what they obtained was a polypeptide chain containing only lysine. However, they were not successful with the poly G synthetic mRNA because poly G tends to hybridize with itself. It actually can form quadruplex. So four strands of poly G mRNA molecules can interact with each other. So they were not successful in getting a poly G mRNA transcript to be translated. Well, it's synthetic, so it's not a transcript per se. Okay, so at this point, we have UUU as the code for phenylalanine and CCC as the code for proline. But what about the other amino acids? So what they did was they had this enzyme called polynucleotide phosphorylase. That's polynucleotide phosphorylase that can actually synthesize mRNA molecules. So the composition of the mRNA molecule that it synthesizes depends on the proportion of the nucleotides present in the reaction mixture. So for example, if you mix 5 portions of um, riboadenosine triphosphate and 1 portion of ribocytidine triphosphate, it will create an mRNA molecule wherein you get a C 1 out of 6. That's the probability. Okay. So if you look at each position, the probability that you get a C at that position is 1 out of 6. And that is again based on the proportion of the riboadenosine triphosphate and ribocytidine triphosphate that were mixed in the reaction mixture, which means you have a high chance of getting AAA in that reaction mixture. And then you also can get AAC or, yeah, AAC, ACA, or CAA. But again, the likelihood of getting these three is lower than getting AAA. And then the likelihood of getting CCA, CAC, and ACC is again much lower. And of course, the lowest probability is getting a CCC in that sequence of in the sequence of that mRNA that you synthesize using this polynucleotide phosphorylase. So they allowed that mRNA transcript to be translated, I mean, the mRNA molecule to be translated, and observe the frequency of incorporation of whatever amino acids were used in the synthesis. So, of course, you will get lysine, right? Because the probability of getting AAA is very high. So the frequency of lysine should be the highest. So the values here are normalized against lysine. So lysine is 100 because it's expected that it should have the highest frequency. And then this is followed by asparagine, glutamine, and threonine. Okay, so for these three amino acids, notice that the assignment of the nucleotide composition of the codon is A2C. So at this point, they cannot say the sequence of the codon. The only thing that they can say is the composition. The codon has two A's and one C. And then we have histidine, which has the next low frequency, histidine and proline. So they assign this AC2, but they also assign AC2 to threonine. And then they assign CCC to proline. Okay. So with this experiment didn't complete the picture, right? But there was another experiment performed wherein, so at this point, they already know about the ribosomes and the tRNAs. So what they did was they have a filter that cannot allow ribosomes to pass through. Okay, so they have tRNAs and the mRNAs. These molecules can pass through the filter, but the ribosomes are retained because the size of the ribosomes are bigger than the pores of the filter. So what they did was they allowed ribosomes to pass through the filter first. So the ribosomes are there in the filter, and then they allowed the passage of the tRNA molecules and the mRNA. Now the mRNA is also synthetic in this case, and what they observed was that there is only one tRNA molecule that gets trapped in the filter. And this tRNA, actually what they used was a tRNA molecule with the activated amino acids. Okay, so for example, here we see phenylalanine. So this is the tRNA for phenylalanine. And then this is the one that was retained in the filter when they used an artificial mRNA sequence of UUU. So this also helped in matching the codon to each amino acid. So with the help of the tRNA molecules, to be more specific, the amino acid or amino acyl tRNA. So when you see amino acyl tRNA, it means you have an amino acid covalently linked to that specific tRNA molecule. Okay. And then by then they were able to establish the genetic code. Okay. So the genetic code is triplet, meaning it has 3 nucleotides. And the sequence of these nucleotides are important. So the genetic code can be presented as this table wherein you read the first letter here, The second letter, you read the first letter by rows, the second letter by columns, and then the third letter by the line number. Or you can use the genetic code presented in a wheel format. So you have in the innermost circle, you have the first letter, and then you have the second letter in the second ring, and then in the outermost ring, you have the third letter, and then you have the corresponding amino acid. So for example, if it's U, so U, first letter, second letter. So you look at this uh, cell of the table. So that's U. If the codon is CGG, for example, so your first letter is C. So you look at the second row here, and then second letter is G. So you should be looking at this part of your table. CGG is for arginine. So in the wheel format, the first letter is C, second letter is G, and then the third letter is G, so it's arginine. That's how you read your genetic code. Okay, so now the question is, if there are only 20 amino acids, why do we need 64 codons? Okay, so first, let's look at the functions of these codons. So these codons, when we say function, it has something to do with how the code is translated. Okay, so we have an initiation codon, we have a termination codon, and we have translation codon. So when we say translation codon, these are the codons that mean or point to a specific amino acid. So the initiation codon or the start codon is AUG. Although if you can note here in our genetic code, this corresponds to methionine. So which means if you start translating an mRNA transcript, the first amino acid that you will place there is methionine. But later on, we will see that the start codon, AUG, is distinguished from an internal methionine, but the codon is still AUG. Okay? Then we have three stop codons, UAG, UGA, and UAA. So once the protein synthesis mechanism encounters one of these stop codons, it's a signal for it to terminate the process. It means you're done with the protein synthesis. And then the rest of the codons correspond to one amino acid. Now we know what reading frame means, but there is one specific type of reading frame that is important when translating an mRNA transcript. So this is what we call an open reading frame. This means you have a continuous sequence of at least 50 codons with no termination codon. Okay? If you encounter such sequence in a DNA, most likely that is a gene. So this information is very critical in genome analysis when you are trying to analyze the genome of, for example, a newly discovered organism. So you're trying to identify which of these sequences are genes. So you have to look for open reading frames. Why is that? Because the probability of encountering a stop codon is 1 in 20 codons. So if you have a DNA molecule, 1 in 20 codons, you will encounter a stop codon. So you need to look for open reading frames. Your genes are usually identified initially through an open reading frame. So you should have at least 50 codons with no termination or stop codon. Now, the characteristics of your genetic code, the first thing that we noticed, of course, is the redundancy. Okay. Uh, 61, 61 of these codons are translated to an amino acid, right? And we only have 20 common amino acids. So we describe that phenomenon as being degenerate or redundant. You have codons that translate to the same amino acid, like phenylalanine has two codons, UUU and UUC. And we can see here in this table the number of codons for each amino acid, and the highest would be 6. Arginine, leucine, and serine have 6 codons for themselves, followed by 4 codons, 3 for isoleucine, and then 2 for most of your amino acids, only 1 for tryptophan and 1 for methionine. So this redundancy is actually very important. Again, we will take a look into this later. So this redundancy does not lead to ambiguity. The codons are not random. Again, this has been optimized through billions of years of evolution. Okay? If you notice in your genetic code, most of the codons that are translated to the same amino acid, they differ only at the third letter. For example, for phenylalanine, they differ at the third position. One is U, one is C. Let's take a look at leucine and isoleucine. Okay, well, leucine, let's take a look at leucine first. For leucine, the first and second letters are the same, except for the third, right? But you also have a different set wherein the first letter is changed from C to U. So that's leucine. For isoleucine, the first letter is also changed relative to leucine from C to A. So why is that? So there is this thing called the wobble hypothesis. Okay. So your tRNA is the adaptor molecule, right? Your tRNA uh, has a certain sequence called the anticodon. So the tRNA, remember, this tRNA is the one interacting with your mRNA transcript. So their directionality should be opposite each other. So for example, here I have the mRNA 5' to 3', that's from left to right. For the tRNA molecule, the 5' is on the right side. So 5' to 3'. So they're anti-parallel. Okay? So there's a segment here in your tRNA molecule which is called the anticodon. The anticodon is the one that is complementary to your codon. Okay? Now, interestingly, so you might ask if there are 64 or 61 61 codons that correspond to 20 amino acids, does that mean the cell also has 61 tRNAs? And the answer is no. So, on the average, there is only at least 31 or 32 tRNA molecules that are actively involved in protein synthesis. So the number is less than that of the codons. Why is that? If you notice again from our genetic code, some of the degenerate codons differ only in the third nucleotide. And that is where the wobble hypothesis enters. Okay. So the wobble hypothesis states that you have the first two bases of an mRNA codon having strong Watson-Crick base pairs with the corresponding bases of the tRNA anticodon, and they confirm most of the coding specificity. So here we are looking at the first and second nucleotides of the codon and the second and third nucleotides of the anticodon. So these two base pairs should follow the Watson-Crick base pairing. If it's a U, then it should pair with an A. If it's a C, it should pair with a G. Now, the thing is, there is actually a different nitrogenous base that you will see in some tRNA molecules, and it's called inositide. Okay, so it's purine. Okay, and this inositide can be found as the first nucleotide of the anticodon in some of your tRNA molecules. This inositide can form weak hydrogen bonds with A, U, and C. So the existence of this different nitrogenous base in your tRNAs, and of course, more specifically in the anticodon of tRNA molecules, further establishes this wobble hypothesis. Okay, so another statement of your wobble hypothesis is that the first base of the anticodon, that is the one that is paired with the third base of your codon, determines the number of codons recognized by that specific tRNA. So, for example, if it's a C or an A, so the first base of the anticodon, if it's a C or an A, it will only recognize one codon, meaning it should follow the Watson-Crick base pairing. So it means you have a G as the third nucleotide of your codon. If it's a C in the tRNA, there should be a U as the third nucleotide of your codon if it's an A in the anticodon. Then further observations, if it's a U or a G, this is less specific, so it may read two codons. To be more specific, if you find a U as the first base of your anticodon, then this could interact with an A or a G in your codon. If it's a G, it can interact with a C or a U. And then finally, which was what was just presented, if it's an I, it can read three codons. So it can read either it can read A or U or C. Which, if we go back to our genetic code, the table, okay, that could mean, and I'm not saying that it's a fact, but we're just applying the knowledge. That could mean CUU, CUC, and CUA for leucine can be read by one tRNA molecule. That is why you have a smaller number of tRNA molecules involved in protein synthesis. So it's not a 1:1 equivalence with your codons. So the wobble comes from this observation that the third base pair in the codon wobbles, so to speak. So the Watson-Crick base pairing is not strictly observed for all tRNA molecules, hence the term wobble. So when an amino acid is specified by several different codons, the codons that differ in either of the first two bases require different tRNAs. So for example, for leucine, UUA will require a different tRNA with CUU because they differ in the first nucleotide. Okay? And then a minimum of 32 tRNAs are required to translate all 61 codons. So 31 to encode the amino acids and 1 for initiation, which is the first methionine residue that is incorporated in protein synthesis. So there's a unique tRNA molecule for that. And then the methionine in any internal methionine, so it's not the methionine that is the start codon, that is a different tRNA is used for that as well. Okay, so now we will go back to the question, why are there degenerate codons? And we will define a bit of terms related to DNA mutation, but uh, this is not the discussion for DNA mutation. We have a separate video for that. So there are types of DNA mutation, and the types of these DNA mutations can be based on several factors or means of classifying them. So you can say you can have transition or transversion mutation if you are looking at how the base is altered. So sometimes the nitrogenous bases that you find in your DNA molecule can be altered. And to be more specific, if a purine nucleotide is changed to another purine, or a pyrimidine nucleotide changed to another pyrimidine. So for example, if it is an A in the original DNA sequence and it becomes G, so that is a purine-purine change, right? So that's a transition mutation. Or if it is a C and then it's changed to T, that's a pyrimidine to pyrimidine transition mutation. And you have transversion wherein the purine is changed to a pyrimidine and vice versa. That's one type of DNA mutation. And then you can also classify your DNA mutation depending on the number of nucleotide affected. So you can have a point mutation. So when you say point mutation, that affects only a single base or a single nucleotide. And it can be insertion, like you insert one nucleotide, but you can also insert more than one, like you can insert a segment, like 10 nucleotides, for example, or you can delete. So deletion is removal of one or more bases. Then we can also classify mutation depending on the phenotype. So the effect of the DNA mutation. You can have a silent mutation wherein it will not change the phenotype. It means you still get the same gene product. The amino acid sequence of the gene product is still the same. You can have a missense mutation wherein a different amino acid will be incorporated in the protein molecule. So that changes the primary sequence of your protein. Or you get a nonsense mutation, which means um, there's a certain codon that was mutated to a stop codon. So that leads to a premature ending of protein synthesis. So you get a shortened gene product. So the genetic code is actually mutation resistant. And the reason why we defined first some of these mutations is for us to understand how the degeneracy of the genetic code gives its resistance to mutation. One thing is that, for example, if you encounter a transversion mutation wherein the C here becomes A, so from a pyrimidine to a purine, that changes the amino acid from leucine to isoleucine, but these two are related, right? In fact, these two amino acids have the same classification. and they have similar structures. So if we look at the impact to the protein molecule, if we get this kind of mutation, there should be very minimal impact. And especially if this amino acid is not part of the active site, it's more of for the architecture of the protein, then there should be minimal to almost no effect. So that's what we call a missense mutation. So you get a different amino acid due to the mutation, right? And of course, due to the wobble hypothesis, that can also give rise to the resistance of the genetic code to mutation. Okay. And yeah, so that is why we have 61 codons for the 20 amino acids. See, these codons are not random. The degeneracy is not stochastic. Okay? And then before we officially proceed to the protein synthesis part, we're going to talk a bit about translational frame shifting. So this phenomenon happens when the ribosome encounters a hiccup during protein synthesis. So what happens is that in the middle of reading the mRNA transcript, the ribosome changes position in a way that shifts the reading frame midway of the protein synthesis. So, like here, this is a gene for a sarcoma virus. So, if you use this GAG, so GAG italicized, so that's the name of the gene. If you use the reading frame for the GAG gene, so you stop here. Okay, so you get your gene product, you get your protein molecule, but if the ribosome does translational frame shifting, so what it does is it will actually move one nucleotide back. So after reading UUA, it will not read UAG, but it will read AUA as the next codon. So it will continue translating and it will get a longer protein molecule through translational frame shifting. And again, this is another proof that the number of genes in a DNA is not equal to the number of protein products. So I guess this also has something to do with efficiency of the DNA, the genome. Like it can get multiple protein products from one gene. Okay. So now we go to the protein synthesis proper. And unlike the first two processes in the central dogma, we will be highlighting 5 stages in protein synthesis. We still have initiation, elongation, and termination, but we will also give importance to the activation of amino acids, and it will be considered the first stage. And then we will also talk a bit about the last stage, which is the processing of your protein product. So, as an overview, um, your ribosome is composed of 2 subunits. So these 2 subunits need to assemble before they can proceed with the synthesis. So after you do the activation of your amino acids, so basically what happens here is that you attach your amino acid to the correct tRNA molecule, and then you assemble the small subunit of your ribosome with the mRNA and the first tRNA molecule. So this should be methionine, right? And then the large subunit also closes the ribosome. So now you have a closed complex, and this is ready for so it's initiated now, okay? And then this is ready for elongation. So in the elongation part, we will also be talking about the mechanism, but the mechanism is just a repetition of itself. So it's a cycle, depending on how many amino acids you are going to incorporate into the polypeptide chain. And then you have termination. So you release the protein molecule from the ribosome, and then these ribosome molecules will do another round of protein synthesis. It can be the same protein product, or it can jump to a different mRNA transcript. And then the protein, of course, as we have learned, undergoes folding. So it has to get to its native state. And then it will also undergo further processing, which again we will talk about. So we will look at the molecules involved at each stage. And I think at this point, you only need to appreciate the amount of molecules involved in each stage. So for the activation of amino acids, of course, you need to activate all of your 20 amino acids. And you have the enzymes that does this. So we call them amino acyl tRNA synthetases. So you need 20 for the 20 amino acids, and you have 32 or more tRNAs depending on the organism. This process is ATP driven because the reason why it's called activation is because you are energetically activating the amino acids for the formation of the peptide bond. And of course, you need magnesium ions for this process to take place. For the initiation, of course, you need your ribosome. So you need the subunits of your ribosome. And this is for the prokaryotic ribosome. You need the mRNA transcript, of course, which has the initiation codon AUG. And you need the first tRNA molecule that has the methionine as the first amino acid. Initiation factors. There are three initiation factors for um, prokaryotes. There are more for eukaryotes. This is also energy driven. So you need GTP instead of ATP, but it does the same function, providing energy to drive the reaction. And of course, magnesium ions. For elongation, you need the functional ribosome, which is the combination of the small and large subunits. And then you need the amino acyl tRNAs that reads the codons and brings the correct amino acid into the ribosome. You need elongation factors. For eukaryotes, you only need one. For prokaryotes, there are three elongation factors, but for eukaryotes, there's only one elongation factor needed. Again, this is also energy driven, but energy in the form of GTP. For termination, of course, you need to encounter the termination codon in the mRNA transcript, release factors, and we still encounter EFG here because it's the one that gives energy for the termination, and IF3 for protection of the active sites of your ribosome. And of course, post-translational processing, aside from folding, of course, you know the folding process. So you need your chaperones and other folding enzymes. Post-translational processing of your proteins will involve a lot of enzymes depending on the type of processing that the protein molecule needs. Okay, so the E. coli ribosome for bacteria, so for prokaryotes, there are around 15,000 ribosomes, and that will account for 25% of the bacterial dry weight, which again shows us the importance of protein synthesis. Now, each ribosome molecule is made up of RNA molecules. That's what we call rRNA or ribosomal RNA, and protein molecules. So it's a complex, it's an assembly not just of protein molecules, but also RNA molecules. And more interestingly, the proportion, okay, you have a higher percentage of RNA molecules than protein molecules in your ribosomes. So the average diameter is 18 nanometers. That is quite big. That is why they can easily be viewed under the microscope. And then there are two subunits, the 30S for the small subunit and 50S for the large subunit. And if they combine together, you get a 70S ribosome. Now, S here stands for the Svedberg unit, um, which is related to the sedimentation coefficient of molecules. So this is when you perform, if you perform centrifugation on them. So like if the molecule has a higher sedimentation coefficient, then it means it needs a higher gravitational force to pellet it out of the solution. That's what the S stands for, Svedberg unit. So if we look at the composition, the detailed composition, there is actually at least only one RNA for each subunit. Like for the 30S subunit of the E. coli ribosome, there is one RNA molecule. And if you isolate only this RNA molecule, it's 16S. And then for the 50S subunit, there are two RNAs, the 5S and the 23S are RNAs. There's still more proteins, but again, in terms of uh, the composition, you still have more RNA because this 16S is big. Okay, so it has a lot of nucleotides for this one strand of ribosomal RNA. So if we look at a more detailed model of the E. coli ribosome, which is here on the left, so that's 50S and 30S, you see that most of your proteins here the proteins are like the noodle like looking illustration. Most of your proteins are found on the surface of your ribosome, and more interestingly, the catalysis is performed by the RNA, not the protein molecules in the ribosome. Okay, so the blobs that you see, those are the RNA molecules. Now, for eukaryotes, it has a similar architecture. You have a small subunit, 40S and 60S for eukaryotes. And if they are combined as a complex, you get an 80S ribosome for eukaryotes. So the cleft here, this is where the mRNA transcript is um, interacting with the ribosome. Okay. Now, the secondary structure of your ribosome, as you can see here, represented as a 2D topology, is conserved across three domains of life: bacteria, archaea, and eukaryotes. So there are only minor differences in their structure. Now, while we cannot appreciate the three-dimensional structure of the three representative ribosomes, this is the small subunit. So this is the 16S or RNA. So while we cannot appreciate the three-dimensional structure because we are only presented with the topology, we are convinced that the structure of your ribosome is conserved because um, overall the structures are similar, right? Okay. So if you want to take a look at the composition of your ribosome, you have the molecular weight there in Daltons. Okay, you can refer to this figure. Okay. So aside from the ribosome, of course, we need the transfer RNAs or the tRNAs for protein synthesis to take place. Again, this is the adaptor molecule that was being thought of by Francis Crick during his time. So the transfer RNAs or tRNAs have four distinct parts. There is the amino acid arm, we have the D arm, the T, psi, C arm, and the anticodon arm. So, as mentioned, there are different or other nitrogenous bases used in tRNAs, like the inositide that we just encountered. So, the D here also uses a different nitrogenous base. So, D stands for dihydrouridine, and you have their psi. The psi stands for pseudouridylate. Then we have general. So the pi there, py or pu, stands for pyrimidine or purine. So it's general. It can be any pyrimidine or any purine that you will find in these locations. Okay. So there are four arms, and overall, the structure of your transfer RNA, the secondary structure is described as a clover leaf like structure because of the four arms. Now, in some tRNAs, they have an extra arm, which is variable in size, but it's quite small compared to the rest of the arms. So, your tRNAs are actually just small RNA molecules, so they vary in length from 73 to 93 nucleotides. So, the longer or the larger tRNAs, those are the ones that are most likely to have the extra arm. Now, if we talk about the three-dimensional shape of your tRNAs, they have actually this bent or twisted L shape. So it is because the T psi C arm is interacting with your D arm, as you can see here. So the T psi arm is represented by color blue and the D arm is represented by the orange. So this L shape was made possible because of this interaction of the two arms. So on one end of your tRNA, that's where you have the linked amino acid, the activated amino acid. And then on the other end of the tRNA is the anticodon. Okay, so let's go to the stages. So let's talk about the first stage, which is the activation of amino acids. Again, the enzyme involved here is what we call amino acyl tRNA synthetase. And there are two classes. So we have the class 1 and the class 2. These two classes differ in the reaction mechanism by which they attach the amino acid to the correct tRNA. So we will start with the first step. So this is for the attachment of an amino acid to a tRNA molecule. There are two stages here. The first stage, which occurs in one active site of your synthetase, is the attachment of an adenosine monophosphate to an amino acid. So this is why you need ATP in the activation stage. Okay, so the carboxyl group of your amino acid attacks the alpha phosphate. Hence, you get an amino acyl adenylate or amino acyl AMP. So from this, it traverses to a second active site in the synthetase. And in this active site, that's when the amino acid gets attached to the tRNA. So it is attached to the 3' terminal nucleotide of your tRNA, which is it is an A. So it's an adenosine. Okay. Now here we see the process branching off because here on the left side, this is the mechanism that shows the class 1 synthetases, and on the right, that's for the class 2 synthetases. Okay, so for class 1, we see here the 2' OH of the 3' terminal nucleotide of your tRNA attacking the carbonyl carbon here in your amino acyl AMP. So AMP is a very good leaving group. So this nucleophilic attack is enhanced. Okay. And then there is a rearrangement because supposedly your amino acid should be attached to the 3' OH of your ribose. So for class 1, the first species that attacks is the 2' OH. But then eventually it gets transferred to the 3' OH. So 2' OH first. So now your amino acid is attached to the 2' via an ester linkage. But we have a trans-esterification wherein the 3' OH attacks the carbonyl carbon again, and then you restore the OH at the 2' carbon, as you can see in this product. This is the final product, okay? The amino acid should be attached to the 3' of the ribose. Now, for class 2 enzyme, which is a different class 2 synthetase, is the 3' OH that immediately attacks the carbonyl carbon. Okay, so now AMP leaves the active site, and then you get your desired product. I'm thinking the difference between the two is probably, of course, in the active site of the enzyme. So maybe for class 1 synthetases, the 2' OH is more proximal to the carbonyl carbon. That is why it is the first OH group that attacks. Versus for class 2 synthetases, it's the 3' OH that is more proximal to the carbonyl carbon. That is why it can directly bind to the amino acid. But that's for a different topic, which we will not be discussing anymore because it's outside the coverage of our class. So, this is the overall reaction. So amino acid plus tRNA plus ATP in the presence of magnesium, you get your amino acyl tRNA and you release your AMP and the pyrophosphate. This is energetically, this is very exothermic. Okay, so it releases around 29 kilojoules per mole of energy. And that is why the reaction is driven forward. Okay. Now, for the amino acyl tRNA synthetases, there's sort of a proofreading process taking place here. Okay, because during protein synthesis in the ribosome, the only thing happening there is the reading of the codon with the complementary anticodon in your tRNA. The ribosome is not checking if the tRNA there has the correct amino acid, which means stage 1 is actually very critical. The creation of your amino acyl tRNA is very important in that you should attach the correct amino acid to the correct tRNA molecule. Okay, so here we see an illustration showing the relative positions of the active sites. So you have the first step, which is the attachment of the AMP to your amino acid taking place on one active site. And then you have the tRNA binding to a different active site. And then you pass on the amino acid from the amino acyl AMP. Now, the proofreading happens because there is a different active site for hydrolysis in case a wrong amino acid is attached to the tRNA. Case in point would be valine and isoleucine. Valine and isoleucine are very similar in structure. They only differ by this CH2, which means if you are trying to attach isoleucine to its corresponding tRNA, but instead of isoleucine, it's valine that enters the active site and is accidentally esterified to your tRNA, your synthetase makes a correction. So again, that correction is made possible by another active site in the enzyme. So it is a hydrolysis site. So if, for example, valine was mistakenly attached to the tRNA molecule, this valine now actually fits this hydrolysis site. So the valine, which is already attached to the tRNA, so there is sort of conformational change. There's a change in position, and valine now is valine enters the hydrolysis site. Okay, and because valine fits snugly into this hydrolysis site, the synthetase will hydrolyze valine. So it will remove valine from the tRNA molecule, and then it will now be ready for another round of synthesis, hoping that it will be isoleucine that will bind this time. So this is a very smart move by nature to provide the correction, the proofreading stage early on, even before you start protein synthesis. Okay, so here is another diagram. So you're doing, you've attached the AMP to your amino acid, um, and then you attach the amino acid. You bind the tRNA. And at this point, the synthetase can check if it has the correct amino acid. If it has the wrong amino acid, then it hydrolyzes that. So it removes the amino acid from the tRNA. So you hydrolyze it. Sorry. Initially, okay, even before the amino acid is transferred to the tRNA molecule, the synthetase is actually already performing some checking at this point. Okay, so again, it has something to do with the fitting, how the amino acyl AMP fits into the active site. So it might accidentally activate the wrong amino acid, but since it checks early on at this point, it can reverse the reaction. So it can release the incorrect amino acid in the AMP. And then another amino acid enters. So again, you have the amino acyl amino acyl AMP. This time, it's correct. So it charges, it transfers the amino acid to the tRNA, and then it can again check at this point. Okay, so if it's the wrong amino acid, then you remove it. Okay, if it's the correct amino acid, then you just release it from the active site. Okay, now this one, the amino acyl, this system of tRNA together with the amino acyl tRNA synthetases sort of comprises a second genetic code. That is because the tRNA molecules are strictly recognized by the correct amino acyl tRNA synthetase. And what I mean by that is there are specific points in your tRNA molecule that are very important to their binding to the amino acyl tRNA synthetase. So this synthetase, aside from checking if it has the correct amino acid, it also checks if it has the correct tRNA molecule. And why are we calling it the second genetic code? Because the tRNA molecule is the one that reads the codons in your mRNA transcript. So at this point, your synthetase plays a crucial role in that it should have the correct tRNA molecule and it should attach the correct amino acid molecule. Case in point, there is the tRNA molecule for alanine. So this is for alanine tRNA synthetase. And this GU base pair found in the amino acid arm of this tRNA molecule is the only feature that is important to its binding in the ALA tRNA synthetase. So why did they say that? How did they establish that this is the only thing important to the binding of this tRNA to the synthetase? So what they did was they made an artificial tRNA molecule, a more simplified version. They simply removed the other arms of the tRNA. So they have the anticodon arm, and they also have the amino acid arm, which contains that GU base pair. And surprisingly, this synthetic tRNA molecule was charged by the ALA tRNA synthetase, meaning it was still able to attach an alanine amino acid to this synthetic RNA molecule. So again, the synthetase plus tRNA sort of makes a second genetic code. And again, stage one is very important for proofreading because again, during protein synthesis, the ribosome is simply reading the codons in the mRNA transcript. So it's trusting all of those synthetases, the 12 synthetases, that they performed their work properly. Okay. Now, when we talked about DNA replication and transcription, we also emphasized the direction of the synthesis. So for DNA, it's 5 prime to 3 prime, and that's the same with RNA synthesis. What about protein synthesis? So for protein synthesis, while you are reading your mRNA transcript from the 5 prime to the 3 prime, and you are synthesizing your protein from the N terminal to the C terminal. And the proof here is the use of radioactive leucine, which is frequently observed in some globin molecules like hemoglobin, myoglobin, etc. So they used red blood cells. And of course, these red blood cells are actively synthesizing hemoglobin. So at some point, T equals 0, they introduced the radio-labeled or radioactive leucine. And when they looked at the complete globin chains, after 4 minutes, they only noticed the radio-labeled leucines near the C terminal. But as they lengthen the incubation time or the time that has elapsed um, before they analyze the globin chains, okay, they get increasingly or progressively longer segments containing radioactive leucine from the time when they introduced the radioactive leucine to the uh, culture. Okay, so this is a proof that it's from the N terminal to the C terminal. Okay, so remember, they analyzed completely synthesized globin chains. So if they analyze these chains early on, like 4 minutes from the time they introduce the radioactive leucine, it means most of the globin chains are almost finished with their synthesis, and then only a few radioactive leucines were incorporated near the tail of the polypeptide chain, and so on. Okay, so now let's go to stage two. So stage two is initiation. So this is not yet elongation. Okay. So for initiation, we need the start codon, which is, do you remember the start codon? The start codon is AUG, which is translated to what amino acid? It's translated to methionine. Now, for prokaryotes, this is not simply methionine, but it is a modified methionine. So what happens is that the tRNA for methionine is charged first. So methionine gets attached to the tRNA first, and then an enzyme catalyzes the modification wherein a formyl group is attached to the amino group of the methionine molecule. Okay, formyl from formic acid. Okay, but instead of a carboxylic acid, you get an amide there. Okay, so you modify. But this is for prokaryotes. For eukaryotes, it's simply methionine that is used as um, the first amino acid, reading the start codon AUG. Okay, so for the initiation step, again, we need the initiation factors, right? So this is the overview, and we will going through each step. So in the first step, you have initiation factors 1 and 3 binding to these sites. Yeah, by the way, there are three sites in your ribosome. We have the A, which stands for the amino acyl. We have the P for peptidyl or peptide, and we have E for exit. Okay, so your initiation factors IF1 and IF3, IF1 binds to the A site and IF3 binds to the E site. So this is to prevent the premature binding of any tRNA nearby. And then what happens next is the binding of the mRNA. So the mRNA binds, and the positioning is such that the start codon or the AUG is right next to the P site. Okay, so how is this, or how does the cell know, or how does the ribosome know? By the way, this is just the 30S or the small subunit of your ribosome. So the ribosome is not yet complete at this point. We have what we call the Shine-Dalgarno sequence, which is upstream of the start codon. This Shine-Dalgarno sequence interacts with the 3' end of the 16S rRNA in the small subunit of your ribosome. And because you have this Shine-Dalgarno sequence, you know that the first AUG that you have there is the start codon. And this is one way of distinguishing AUG as a start codon and any internal AUGs in the mRNA transcript. Okay, so the start codon AUG should be very near the Shine-Dalgarno sequence. And then the tRNA with the formyl methionine, the modified methionine, plus this initiation factor 2, which has a bound GTP, enters the complex. So they become part of the complex. The tRNA enters the P site. So the anticodon here should be complementary to the start codon. Okay, and you have the initiation factor 2 there interacting with the small subunit of the ribosome. And then finally, the large subunit of the ribosome caps the small subunit. So now the complex is complete, and with this, all of the initiation factors are released, and they are released using energy. So recall that IF2, initiation factor 2, has a bound GTP. So it gets hydrolyzed into GDP plus a phosphate group, and that releases energy, which drives the release of your initiation factors. So now you have the initiation complex. And that is the end of the initiation stage. Now, for eukaryotic initiation, so there are three initiation no. There are more than three initiation factors for eukaryotic protein synthesis, but there are initiation factors that are analogous to IF1, 2, and 3 of prokaryotes. There is an initiation factor that helps guide the mRNA to its correct position, but you still have that Shine-Dalgarno sequence, or the counterpart of the Shine-Dalgarno sequence in the mRNA transcript. And the removal of the initiation factors and the formation of your initiation complex is also energy driven. As you can see there, the GTP. This gets hydrolyzed. It releases all the initiation factors, and then you form the initiation complex. Okay. Now, the counterpart here, this Scheindel-Garno sequence, it's not the Scheindel-Garno sequence, okay? It's a counterpart. So what happens is this initiation factor interacts with the 5' terminal of the mRNA transcript, and this guides the mRNA transcript to the site, okay, more specifically in the proximity of the E site. And then what happens is that the ribosome will slide itself along the mRNA transcript until it reaches the start codon. Okay. Now here we see some representative sequences of the Scheindel-Garno sequence. Okay, so you see the relative position of this sequence to the start codon. It's very near to the start codon. Okay, so it's rich in G and A. And here you can see the complementary sequence found at the 3' end of the 16S rRNA of the small subunit of the ribosome. Okay, so now we are in the elongation step. So this is an overview. Again, the process repeats itself as long as there are amino acids being added to the growing polypeptide chain. So let's look at the first picture here, which is the initiation complex basically. So what happens here is that the A site is empty, right? So the A site will now look for the tRNA that has the anticodon complementary to the second codon, the one after the start codon, AUG. So this tRNA has a bound protein called TU. It's EFTU actually to be more exact. It's an elongation factor. It's EFTU with a bound GTP. So this guides the binding of the correct tRNA to the A site. So you have there the second amino acid of your polypeptide. So what happens here is once it has correctly positioned the tRNA, the elongation factor will remove itself, and this removal is also driven by energy. So it needs to hydrolyze the GTP there to remove itself. And then what happens here? Once you have the second amino acid there, then the synthesis takes place. Remember, the synthesis is catalyzed by the RNA of the ribosome. Okay? So now we have the two amino acids there close to each other. So how are they connected? So how is the peptide bond formed? Let's look in detail. So what happens is the amino group of the second amino acid attacks the carbonyl carbon of your first amino acid. Okay, so remember this is attached to the 3' carbon of the ribose. So it attacks this carbon. Then there's the electron rearrangement to the oxygen. Now this oxygen, it becomes negatively charged. But what happens is it will rearrange itself. So it will become pi electrons again. But instead of this bond will now this bond will be broken. So it will abstract a proton from this sugar. And then this sugar will now attack this proton. Okay. So essentially, we have a peptide bond. So this becomes a carbonyl carbon, and then you have the peptide bond, and then you have the amino group there. Okay, so this oxygen bound to this alpha carbon of your first amino acid. Sorry, no, this is the alpha carbon. It's bound to the carboxyl carbon. Okay, this oxygen, this bond will break. Okay, so it will abstract this hydrogen from the 2' carbon of the ribose, and then this pair of electrons will abstract this hydrogen, which means you restore the ribose, right? You end up with the 2' OH and the 3' OH. And the ending is you now have the dipeptide attached to the second tRNA, which is still in the A site. Now, when this happens, the two tRNA molecules will change their structure a bit. So there's a so-called hybrid structure because part of your tRNA is still in their original sites, but another part of them has moved to the adjacent site. So the first tRNA is still in the P site, but its amino acid arm is already in the E site. Likewise, the second tRNA, it's still in the A site, but its amino acid arm is now in the P site. So what will happen here? So what will happen is a translocation of your ribosome. The ribosome needs to move along the mRNA transcript. And so this is our current status. This needs an elongation factor, a different elongation factor, EFG, which is also again bound by GTP. So this EFG has a high affinity to the A site. So it will sort of compete with the second tRNA that is currently occupying the A site. So what will happen is um, it will force these two tRNA molecules to move. No, not exactly, but what will happen is it's the ribosome that will move. So remember, your tRNA molecules they have interactions with the mRNA transcript. So imagine your tRNA molecules are static in position. It's the ribosome that will move. So what will happen is if the ribosome moves, the second tRNA is now in the P site and the first tRNA is now in the E site. And it is the EFG currently occupying the A site. So remember, the EFG has some affinity with the A site. So this translocation of your ribosome is driven by energy. So the GTP in EFG is hydrolyzed, and that provides the energy for the translocation or the movement of your ribosome by one codon along the mRNA transcript. So the EFG is removed from the A site, and now you can repeat the process. The tRNA in the E site will dissociate, so it will now exit, and then you now have an empty A site ready for another round of peptide bond formation. So the process will repeat itself until your ribosome encounters a termination or a stop codon. So we are now in the termination stage. So for the termination stage, you have the polypeptide chain there. Remember, you are growing the polypeptide from its C terminal, and you always find the last tRNA molecule in the P site. Okay, so for the termination stage, we need release factors. So your release factor will bind to your UAG, to your A site. So now your A site is facing the stop codon. Since there are no tRNAs that recognizes this codon, this stop codon, it's time for the release factor to play its role. So it will bind to the A site. Okay. And then this binding catalyzes or signals the release of your polypeptide chain. But how does it do that? So, what actually happens here is that if you have the release factor bound to the A site, there is another hydrolysis. No, sorry, there is another condensation reaction taking place. But instead of connecting this polypeptide chain to another amino acid, it will only be condensed with a water molecule. And that is catalyzed with the help of your release factor. So now you release your polypeptide chain, and then you need again energy to dissociate all of the molecules that are currently interacting with each other. So you have EFG again. You will use this energy, GTP loaded EFG, to drive the dissociation of the large subunit and the small subunit. And you again need IF3 to cover the E site in your small subunit of the ribosome. And that's it. That's how you terminate your protein synthesis. Now let's talk about efficiency. Again, although your cell synthesizes proteins at a slower rate, like we saw the processivity, right? It's 100 amino acids in 5 seconds. Uh, it can still produce a lot of protein molecules from the same mRNA transcript because one mRNA transcript can actually be translated by multiple ribosomes. And that is what we call a polysome, which is again, this can be observed under an electron microscope. So polysomes, it's like a bracelet. Like you have the nylon. The nylon would be your mRNA transcript, and the beads would be your ribosomes. So what happens here is that multiple ribosomes are actually translating the same mRNA transcript. And in the context of your bacteria, this can also take place during transcription. So even while the mRNA is still being transcribed, like it's still being synthesized, the cell can actually start the translation of the protein product. So you have one ribosome starting the process. And once it has reached a certain length of your mRNA, a second ribosome can actually start synthesis again, and so on, which means you create multiple protein products from the same mRNA transcript in a short period of time. So that is one of the efficient ways by which the cell compensates for the low processivity rate of protein synthesis. Okay, and then we go to stage 5, which is post-translational processing of your protein molecules. So, of course, your protein molecules need to reach their native state. So, they have to fold properly. In some cases, they can be helped by your chaperone proteins and other folding enzymes or folding proteins. Aside from that, some processing can also take place, such as the removal of the N-terminal amino acid residue. Like in the case of prokaryotic protein synthesis, they remove the 4-male methionine. In eukaryotes, they can also remove one or more of the N-terminal amino acid residues to get the final protein product. In some cases, the N and C terminal amino acid residues can be modified. For example, you can modify the N terminal with an acetyl functional group. So there is an enzyme that does this. And then this modification has a specific function as well. There's also loss of signal sequences. So most proteins, when they get synthesized, they contain this so-called signal sequence. And this is the signal for the cell to be able to transport them to their correct location inside the cell. Like Should they be in the cell membrane? Should they be in the chloroplast, for example? Or should they be in um, the Golgi apparatus, in the nucleus, and so on? So once the protein has reached its destination, its supposed or right location inside the cell, it can be processed by removing the signal sequences. So in most cases, these signal sequences are 15 to 30 amino acids long, and they are located at the N terminal of your protein molecule. Next, you can modify any of your internal amino acid residues. When we say modification, we are actually adding functional groups to these amino acid residues. We can add phosphates, we can add some carboxylates, and we can also methylate some of these amino acid residues. And some of these modifications change the landscape. Like, for example, if you add phosphates, you are actually adding negative charges. And again, this has something to do with the function of your protein. So these modifications are not random, they are important to the function of whatever protein you are modifying. If you are familiar with glycoproteins, so the attachment of your carbohydrates also take place after translation, after you synthesize your protein product. So this is important in blood typing, for example. So the what do you call this? You have the antigens, right, on the surface of your red blood cells. So the structure, those are actually glycoproteins. So they are proteins with carbohydrates, like short segments of carbohydrates attached to them. And the structures of these carbohydrates determine whether it's an a B. If you have none, then that's an O, or if it's an AB. So when we study carbohydrates, we will be learning about that blood typing. Okay. And then, of course, you are already familiar with the functions of your proteins. So, proteins with prosthetic groups need their prosthetic groups. So, after translation, you need to add their prosthetic groups, right? Or their cofactors, coenzymes. Okay. Some proteins can actually undergo proteolytic processing. So, there are proteins that, when translated and folded into their native state, they are still in the inactive form. This is the most common example would be enzymes. So, like for example, the inactive pepsinogen. It's inactive because its active site is actually masked. So, some protease will remove the mask of the active site, forming the active form of the enzyme, pepsin. So this is also another type of processing after translation. Of course, your proteins, if they have disulfide bridges, this happens after translation and now. So those are the typical or common post-translational modifications that happen to your protein products. Now let's try to work a very simple example on how to crack the genetic code. So we have here a single DNA strand. So it says complete the other half by writing the complementary base pairs. So let us write the complement. So that's T, A, C, G, G, T, A, T, A, C, 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 A, T, and T. Okay, so this is the complement. So that's A, T, T, A, G, C, C, G, C, G, A, T, T, A, A, T, T, A, G, C, G, C, G, C, T, A, A, T, A, T. So we have the complement. Now, although the directionality is not written, if it's not written, then we can assume that for the given strand, it's written from the 5 prime to the 3 prime, which means the complementary strand that we wrote is running from its 3 prime down to its 5 prime. So now it says we just wrote in the template strand of the DNA. In that case, it means the given is the non-template strand or the coding strand. And what we wrote is the template strand. So now we will be using the template strand to transcribe a strand of mRNA. So again, they should be anti-parallel. So since the template strand is already written from 3 prime to 5 prime, then we can write our mRNA transcript as is. Actually, what you can do is just copy the sequence of your coding strand, but your T should be changed to U. So it's G, C, C, A, U, A, U, G, 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 U, A, 3 prime. Okay, so we have our mRNA transcript. We can also check the complementarity of this with the template strand. So that is, so if it's T, that should be an A. If it's A, it should be a U. C, G, G, C, G, C, T, A, A, U, T, A, A, U, C, G, C, G, C, G, A, U, T, A, T, A. Write down the tRNA anticodons that pair with the mRNA strand. So for this one, it helps, like in my case, personally, it helps if I identify the triplets. Okay, which means that for the first codon, the anticodon is, so from 3 prime, the anticodon is UAC. This is for the first. This is the one, this is the anticodon for AUG. And then for the second codon, from 3 prime, that's GGU. For the third anticodon, 3 prime, that's AUA. For the fourth anticodon, 3 prime, that's CCC. And for the last codon, so the fifth anticodon would be AUU. Okay. Now we are going to use our genetic code. Now, when you translate, you read your mRNA, not the anticodons. So for example, so from the N terminal, our first amino acid is methionine, followed by the second codon is CCA. So it's CCA, that's proline. The third is UAU, UAU, tyrosine. The fourth codon is GGG, that's glycine. And of course, the last codon is a stop. So what we get is a 4 amino acid peptide. Okay. So in this case, we will not be specifying that it's a 4-mil methionine. So we will simply be translating the mRNA transcript to the corresponding amino acid sequence.